Well, good afternoon, everyone. I know there's lots of people joining by Zoom, and we're also recording this, and we have folks in the room, and others will probably join as well, but I want to welcome all of you to a special NHGRI Directors Research Seminar, which I don't know if I ever did one of these or not, but I invited Matt to come visit, and I wanted him to give a talk, and it wasn't part of a regular seminar series or anything, so I just said, we're just going to call it a special director special. Seems like something I should be able to do, so I did it. So welcome to those in the room, those joining virtually. It's wonderful. We have a special guest here at NHGRI and at NIH today, Professor Matt Hurles. Matt's a fairly 6 months in, right? 6 months into being the newly appointed director of the Wellcome Sanger Institute in Hingston, UK. Keep in mind, that's an amazing research institution that has had some real luminaries in genomics luminaries in genetics and genomics, starting with Nobel laureate John Sulston, I should point out. Prior to this role, Matt was the head of the human genetics program at the Sanger Institute. He's a well-respected human geneticist. He studies the causes and consequences of new mutations that occur in DNA as it's passed from one generation to the next. He leads a research group that's focused on deciphering the genetic causes of severe developmental disorders. I'm certain that's what he's going to talk about in part during his talk. And he is best known for his work on characterizing the extent and impact of structural variation of the human genome and on deciphering the genetic architecture of severe neurodevelopmental disorders. He originally trained in biochemistry at University of Oxford, obtained his PhD in genetics at the University of Leicester, and he's an honorary professor in human genetics and genomics at the University of Cambridge and a fellow of the Academy of Medical Sciences. Notably, in 2019, he was elected to the Royal Society, which is an independent scientific academy of the UK dedicated to promoting excellence in science for the benefit of humanity. So please welcome me and joining Matt, who's going to give a talk on deciphering developmental disorders. Matt.
Thank you very much, Eric, and hopefully everyone can hear me okay online as well. So, and I've already learnt my first lesson, which is if I want to get something done as a new director, I just need to call it special.
That's right.
Yeah, excellent. So that's a good tip. So, so it's a great pleasure to be here. It's my first time on the NIH campus. And I'm not going to speak very much from a Sanger Institute perspective, very much my own personal kind of research perspective. So I'm going to talk to you about this deciphering developmental disorder study, and I know many of you think about some of these same kind of genomic medicine kind of topics as well. And I think one of the things I'd like to get across is the value of doing these things at scale and learning from doing these things at scale about general principles that help us to understand where to go next, because we haven't by any means solved all the problems that we need to solve. And then on the second half, I'm going to talk to a thing that is the bugbear of any of us doing genomic medicine, is the variance of uncertain significance problem. And this is something which I think is yielding to new technologies, and there is a real opportunity for a kind of collective international endeavor on this. And I'll give you a deep dive vignette into our kind of one of our kind of most recent experiences on this and why we think it's so valuable. So for those of you who aren't familiar with it, the Deciphering Developmental Disorders Initiative, or the DDD study, was established in 2010. And was very much a collaboration between all of the sites in the UK and Ireland who care for clinical genetic sites who care for children with severe developmental conditions, 24 of them, over 200 clinicians. And it was really about trying to take the genomic technologies that we had access to in the research domain that weren't currently in the clinical domain and see how we could apply these. So initially microarrays at higher resolution, but then moving into sequencing. And so it was very much a twin-track project from the outset with clinical deliverables in terms of diagnoses back to patients, but also research deliverables in terms of understanding the fundamental genetic architecture of these conditions. And so over the course of the next 5 years, we recruited over 13,000 families, all systematically phenotyped by their clinicians, over 200 different clinicians. And then we applied these technologies, which to date is largely exome sequencing and array comparative genomic hybridization, but I'll talk predominantly about the exome sequencing, often in the context of the family, so sequencing both parents and children, and then feeding back possible diagnoses back to the clinical teams who have the richness of data at their fingertips to interpret those and see whether they are truly diagnostic or not. And currently, we're diagnosing about 40% of these children. That has grown, as I'll show, over the course of the project. It didn't start off at 40%. So this is very much a clinical population at need of diagnosis, rather than something more systematic in terms of a kind of research study with very tight inclusion and exclusion criteria. So it's quite heterogeneous, but by far the biggest phenotypic contributor is neurodevelopmental conditions, with over 80% having neurodevelopmental conditions, often intellectual disabilities, developmental delay, but often with structural abnormalities or behavioral neuropsychiatric phenotypes as well. We also, and this will come important in a bit, captured some of the potential for prenatal risk factors during pregnancy. And just to give you a sense of the nature of the families, in most of these families, the child who's affected is the only affected member of the family. So these are not big pedigrees. These are typically 2 healthy parents and 1 sick child. So back in 2010, when we initiated this project, the informatics of how to do this was somewhat in its infancy, and we ended up having to build this system that was somewhat more complex than the average kind of research project. The average kind of human genetics research project might involve bringing in some samples, sequencing them, calling some variants, doing an analysis, and writing a paper. And that's undoubtedly part of what we're doing here. But we also have important front ends to the clinical community. We're capturing clinical information across 24 sites. And we're feeding back at the end of the process potentially diagnostic variants to those clinical teams to try and make diagnoses for their patients. And in the middle, the real learning part of the process is these steps here, where we're trying to filter down the very large numbers of variants we discover into those that might be worth feeding back to clinical teams, and then seeing how well we're doing, learning from that, changing the filtering, and so forth. And so there's actually turned out to be much more iterative than I think we originally anticipated. And this clinical front end that we were using was leveraging an existing portal known as Decipher, which my colleagues had set up back in 2004 to enable sharing of limited genotype and phenotype information on undiagnosed children to try and identify matching children in other parts of the world. So it was an interface that clinicians were used to looking. And if there's one thing I've learned, is if you want clinicians to look at data, you need to take the data to where their eyeballs already are and not expect them to look somewhere else. So the big risk, of course, with our NHS colleagues was that they were going to recruit a patient and we were going to give them back 20,000 variants. And that was something they were very fearful of happening. And so to build trust with them, we built a system which would involve the maximum amount of computational filtering, and then only sharing with them the variants that were plausibly diagnostic. And so, as we all know now, but was less clear then, we've all in this room got 20,000 variants in our genes. The vast majority are common. There are only a few hundred which are both likely to be damaging to the gene and rare enough in the population to be plausibly a cause of a rare disorder. And then we cross-reference that against genes that are sufficiently robustly associated with disease that they can be used diagnostically. And as I'll show, that changes quite dramatically over the past 10 years. We integrate that still in anyone in this room will give us 10 to 20 variants, so quite a lot of diagnostic interpretation potentially. And then we use the parental DNA sequence to filter out those that are inconsistent with what we know about Mendelian inheritance. And so it ends up with only the clinician needing to look at a handful of potentially diagnostic variants. And we started off deliberately conservative when this to build trust with our clinicians that we weren't going to dump 20,000 genetic variants on them. And as it turned out, we didn't actually move that far away from that initially conservative position over the course of the 10 years. So this is a very complex picture of kind of 10 years of bioinformatic evolution, if you will, and really reflects the fact in this study we had 3 main types of data: exome sequences, array CGH data, SNP array data. And out of those 3 data sets, we wanted to infer as much genetic variation as possible. So lots of structural variants of different flavors. And we ended up with over 10 different algorithms being applied to these data sets to try and call these genetic variants. But ultimately, to do clinical interpretation, we want to bring them together. We wanted to build a system where we could identify a patient with a recessive condition if they had a SNV that had been identified from exome sequencing, but a deletion on the other allele that we only picked up on the array. So to do that, we had to not only call these data, we had to actually combine them together in one source. So this is multiple of these algorithms we developed ourselves to try and tackle these problems. And I'll say in a little bit quite how much we got from each one of these different approaches. But fast forward now to earlier this year and work that Caroline Wright, one of the leads on the study, has kind of pulled together. Having now analyzed data on these 13,000 families, what do the results look like after 13 years of effort? So what I'm showing you here is the number of variants that actually ended up getting fed back to the clinical teams. So there's 13,000 families. We've fed back to date about 20,000 variants. So that's actually on average 1.5 variant per family. So pretty conservative, as I mentioned. And actually some of those families, especially those families where we've got DNA from both parents, so we've got a full trio, neither parent is affected. Actually, in 3,000 of these families, we fed back precisely 0 variants to interpret. Whereas if you look at individuals who didn't have DNA available from both parents, so that they're here known as singletons in blue, you can see it was an average of 2, 3, 4 variants that got fed back. So pretty conservative compared to many of other approaches. But we don't think we're missing a great deal. So this is then how it's tracked then over the last 10 years or so. So the first thing to note, in blue, these are the number of genes which we could use diagnostically because there was enough evidence of them being associated with developmental disorders. So it's about 800 back in 2012. It's almost 2,000 now. So it's almost doubled the number of genes that one can diagnose. Over the course of that time, the data sets that we've generated in the dashed line, as we've increasingly sequenced a greater proportion of the cohort, have increased. And so now we've returned back about 20,000 variants and made about 5,000 diagnoses. And you can see, so many of these individuals, especially the ones sequenced at the very beginning, will have been reanalyzed 6 or 7 times. And many of those diagnoses that are made at the very end of the process are in individuals that have already been analyzed many, many times, but it's only recently that the gene in which their diagnostic variant has been found has become sufficiently robustly associated to be used diagnostically. So what this really points to is the real importance of reanalysis of existing genetic data to maximize diagnostic yield. And one of the disappointments is that in certainly most of the healthcare systems I'm familiar with, it's been very hard to engineer this reanalysis into standard clinical practice, even though nobody that I'm aware of actually contends that this is actually robust evidence that reanalysis is for patient benefit. And I think that speaks somewhat to some of the difficulties in implementing even quite rational, well-supported new ways of working. So to give you a sense of those 20,000 variants and that we got fed back that to be interpreted, what different algorithms did they come from? Well, the vast majority were SNPs and indels and were picked up by those small variant detection methods. But each one of these different structural variation algorithms did pick up a handful. And even the lowest yield that we've had here, an algorithm to pick up mobile element insertions, there are 4 families out of 10,000 where the diagnostic variant was a de novo mobile element insertion into a developmental disorder associated gene that would not have been picked up if we hadn't run the algorithm. But there's very few clinical services that I'm aware that run this kind of range of algorithms. It just goes to show the difference between different approaches is not just driven by what data you're generating, but also how hard you work on those data to mine out the variation within it. So this then is going back to, so what did the clinicians then decide about those 20,000 variants? And if we start on the right-hand side, so there's different classes of variants that we fed back under different types of inheritance, different Mendelian patterns of the associated gene. And you can see, and they're colored according to the clinical interpretation, with red being pathogenic and likely pathogenic. And you can see that there's some classes of variation we fed back which have a very high likelihood of being the underlying diagnostic cause of their disease, and others that had a much lower likelihood. And unsurprisingly, the ones that have a much higher likelihood are the ones that occurred de novo in the child, and the ones that have a much lower likelihood of being interpreted as being a kind of the pathogenic variant are ones where were either inherited or of unknown inheritance. And if one looks back on the left-hand side, we can see that the majority of these red, these pathogenic and likely pathogenic variants are de novo, whereas the majority of these VUSs in this kind of stippled pattern were the variants of uncertain inheritance. So this kind of pattern of the importance of de novo mutations in disease is true both for the study that I'm talking about in the main, this postnatal developmental disorders study, where about over 3/4 of the diagnoses are de novo mutations. But in a separate nationwide study that was a bit smaller, looking at prenatal structural anomalies picked up by ultrasound, which again involved a big network, nationwide network of fetal medicine centres, had a much lower diagnostic yield, so only about 9% of we could find a monogenic diagnosis for. But again, the majority of those were de novo mutations. Now that raises the question, have we got such a lower diagnostic yield prenatally because these are fundamentally less monogenic conditions? Or is it that there are large numbers of genes that cause prenatal developmental disorders that we haven't yet discovered? And I'll come back to that in a moment. So this essentially is after 13 years of effort where we are. We've managed to diagnose about 40% of the kids. About 3/4 of those are de novo mutations. About another 1/5 to 1/4 have a variant of uncertain significance in one of these known developmental disorders associated genes. And about 2/5 don't have a diagnosis and don't have a VUS. So this actually provides us then, given that there is this pie chart represents 13,000 families, quite a powerful approach to now, having brought the data together in bulk, to analyze what factors predict whether someone was likely to get a diagnosis or not. And that's what this rather busy slide purports to show, and I'll just walk you through it. Essentially what we're looking at is different variables down the left-hand side that we kind of collected and knew about at the outset of the study. Some of them are demographic, some of them are about prenatal risk factors, some of them are phenotypic, some of them are just about the family history. So, and then what we're looking at is we're saying, did any of these factors in a multiple regression analyses, were they associated with being less likely to be diagnosed or more likely to be diagnosed? And essentially what this says is if you're more likely to be diagnosed if the child was female rather than male, about 55% of the cohort is male, 45% female. If the child had lower levels of homozygosity, and we can come back to why that might be. If the child was recruited as a trio, it makes a big impact. So if DNA is available from both parents on the likelihood. If the child is the only family member affected, and indeed what we see is the more affected family members there are, the less likely one is to get a genetic diagnosis. The more severe the neurodevelopmental disorder, the more likely to get a diagnosis. And intriguingly, the more prenatal risk factors there were, the less likely to get a genetic diagnosis. So it's certainly not the case that none of these children had genetic factors. In fact, still even within the extremely premature children, there was still a significant fraction where there was an underlying genetic cause. But whether the prenatal risk factor was prematurity or maternal diabetes or maternal use of anti-epileptic drugs, all of these were associated with a lower diagnostic yield. So it's only really putting these data together that one can start to tease out these factors. And the other part, again, concerning from an equity perspective, is on ancestry. If a family had African ancestry, they were less likely to get a diagnosis. Now, one thing we can do is tease apart that signal of African ancestry and subdivide it between those recruited as trios and those recruited without DNA from both parents. And we can actually see that African ancestry families recruited as trios have the same diagnostic yield as other trios, whereas it's the African ancestry individuals who didn't have DNA from both parents available that are really suffering from lower diagnostic yields. And we know that part of this is due to the nature of the reference data sets, but even part of it is likely due to the underlying greater genetic diversity in Africa. The haystack is bigger, the needle is the same size. So we get a couple of messages that I think are important out of this. One is, the current way we do diagnostics, this is, I think, reasonably reflective, is underserving those of African ancestry. It's also underserving single-parent families. And I think we should bear that in mind. I'll come back in a little bit as to into other dimensions of equity. So one thing that we didn't from the outset of the project do was build in a systematic approach looking at outcomes of receiving a diagnosis and what happened. So recently, Caroline Wright and colleagues have kind of retrofitted this onto the study and managed actually to, working with the clinical teams who have given these diagnoses back to over 4,000 of the families, pulled out what actually the implications were for, on the clinical side. So you probably can't read the details, but if I summarize in light blue at the top, it indicated that there were treatment changes in about 2% of families that received a diagnosis. So a low proportion, but that's not surprising given the nature of the disorders that we're looking at and the low proportion of them being treatable. But about a third received additional clinical care in the forms of referrals to other clinical specialties for screening in the green across a whole range of different specialties that wouldn't otherwise have occurred. A large proportion, kind of well over half, received disease-specific information and support. One of the things we did as part of the project was when we discovered new conditions or found that conditions were particularly prevalent, we tried to put together family groups and clinicians to write up information leaflets so that families would have more information than they currently had about their condition. About a third received reproductive counselling, and of that, and actually about 2% of the cohort in general actually had reproductive testing as a result of that, either prenatal diagnosis or pre-implantation genetic testing. And there were some adverse events in about 0.5%, often around significant anxiety caused by the diagnosis itself or the prognosis for the future. So this is by no means a perfect study of clinical outcomes. It will no doubt differ based on the clinical population and indeed the healthcare setting, but I think it gives you a flavour of the different ways in which diagnoses have impacted families and their clinical care. But that's the 40% for whom we could get a diagnosis. What about the remainder for which we haven't been able to get a diagnosis? And where do we need to look next to try and find those diagnoses? Well, I think it's fair to say we should question right from the very beginning, is there something even genetic to discover here? Could there be underlying environmental causes? Could there be non-monogenic genetic causes? But then if we think about there being monogenic causes that we haven't yet discovered at the bottom, we can broadly speaking break these down into three groups. So where we haven't discovered the variant, and therefore we need to run a different assay or a different algorithm to identify the variant. And where we have discovered the variant, and that variant is either in a known developmental disorder gene, but we don't have enough evidence to say it's pathogenic, and so it's a variant of uncertain significance, or a VUS. Or it's detected, but it's in a gene that doesn't currently have enough evidence to use it diagnostically. And because we can, I won't go into the details, but we can look at the data that we have in aggregate, and by comparing the array data against the exome data is one thing that helps us speak to this first point here. My best guess is that maybe single figures percentages for families will be solved by running different assays or different algorithms. I think we've got decent evidence that actually, well, as I showed you before in the VUSs, there's 22% of the families have a VUS, the majority of which will probably turn out to be benign. But we have a good reason to think that actually there are many more developmental disorder genes still to be discovered. And therefore, we're putting our effort very much on the gene discovery side of things, more so than applying different algorithms or different assays. So one of the kinds of evidence that we use is taking these data in bulk, looking at a particular class of variation to try and understand whether it's involved, and just asking, do we see more of that class of variation than we'd expect to see by chance? What's the overall burden of that type of variation? And then having identified for a particular class of variation, for example, truncating variants in protein coding genes, ask the question, how much of that variation, that excess, is accounted for by genes that we already know to be associated with neurodevelopmental conditions versus ones that are novel? And when we do that, looking at protein coding de novo changes, we can see that we're only explaining about half of that excess. So 20 to 25% of children probably have a de novo mutation as their cause, but they're in genes that we haven't yet associated with disease. So that really pushes us in the direction of gene discovery. The other thing that we can do with these kinds of bulk analyses across many hundreds or thousands of patients is go back to answer that question that I posed earlier. Do we see a lower diagnostic yield prenatally in the PAGE project because there's lots of genes we haven't yet discovered, or is it that it's fundamentally less monogenic? And so if we look exome-wide at the excess of damaging variants, we can see that excess is considerably lower for missense and truncating mutations in the prenatal project than it is in the postnatal DDD project. So we think it's much more likely these are fundamentally less monogenic traits, and there's more diverse etiologies than there's a large number of as yet to be discovered prenatal specific developmental disorder genes. It's also worth noting that when we do these kinds of analyses to say how important is a particular class of variation, it clearly depends from population to population. And so one of the interesting questions we had for a long period of time is how much undiscovered recessive disease is there in our population? And the answer actually is pretty low. And you can see on this left-hand plot for the majority of the kind of UK population of British European ancestry, there's 10 times more causation from de novo mutations than recessive disease. Whereas if one looks at the British Pakistani subgroup, it's more like half and half. And if one splits that British Pakistani subgroup into those that have related parents and those that have unrelated parents, those with unrelated parents look just like the left-hand plot. So it's not really a property of ethnicity so much as parental relatedness. But it does speak to the fact that whatever we identify in terms of genetic architecture will vary between populations. And we're doing a study at the moment with NIH funding in collaboration with partners in Africa called DDD Africa. And we are still putting together the data. But I think it's highly likely we'll see a pattern that looks very much like this left-hand plot here, because de novo mutations are coming up again and again as the diagnoses that they're making. So focusing on de novo mutations, thinking about how do we do gene discovery better, having taken 4 years to recruit across our entire country as many patients as possible, we still hadn't addressed the problem. So we're clearly going to have to aggregate data together internationally. And a few years ago, we put together a collaboration with GeneDx, a US diagnostic company, with Radboud University, who are probably the leaders in continental Europe at diagnostics in Nijmegen, on a study on 30,000 trios and identified evidence for about 25 or so new disorders, which gave diagnoses in several hundred patients that wouldn't previously have had them from those families. And we're now repeating that analysis now with about 80,000 trios, so with somewhat mind-boggling, almost 250,000 exome sequences in one disease study, by pulling together additional data from those 2 clinical centres, but also from the diagnostic testing now going on in the UK, now being done by Genomics England and the NHS. And I don't have some firm data on this, but we are very much looking at, of the order of 50 to 100 novel developmental disorder-associated genes after this analysis, and probably over 1,000 patients who will get diagnoses as a result of discovery of those genes. So it's just important to note that this is, international collaboration is critical here, but what's also critical here is that data that's being generated largely for clinical purposes is being made available for research. This gene discovery wouldn't be possible if this data got siloed away and was no longer available for research. So it's very much kudos to Genomics England, GeneDx, and Radboud University for making that happen. And that is something that is a fear of mine, is that as genomics goes into medicine, and yet we don't have enough answers for patients, that we don't make that virtuous loop of having the data come out in kind of trustworthy ways to enable to be used for research to actually make best use of those data. This is as true in diagnostics as it is in global pathogen surveillance, as it is in essentially every other domain of genomic medicine. So one thing I just want to go back to is this idea of equity. And we don't have data within this on our own cohort, but a really intriguing study recently came out of the UK on Imagine ID, which is a national cohort of children with intellectual disability of genetic origins. And they did a study where they looked at the diagnostic variants that were found in children, and they subdivided it into, this is the index of multiple deprivation. So you've got the least deprived quintile here, the most deprived quintile here, and in the least deprived quintile, what you're seeing is that the majority of the diagnostic variants in children were de novo, whereas in the most deprived quintile, the majority of the diagnostic variants were inherited from a parent. Oftentimes, that parent had not been annotated as clinician as having had a similar condition to the child. So we don't have the data to test this in our cohort, but a key thing here is that we may well be underdiagnosing families in lower socioeconomic groups. So as well as that single-parent family, African ancestry inequities, we may be looking at a socioeconomic equity here as well. It does raise the potential that there are individuals with damaging genetic variants who are maybe more resilient, and we really like to understand that better. And it does also raise the potential in terms of gene discovery. Focusing on de novo mutations may well be missing some of the pathogenic variants. We may be able to gather more evidence by combining data across different modes of inheritance. And so Caitlin Samoka, who did a postdoc in my group, is now running her own group at MGH in Boston, has done some work to test this out, looking at these inherited variants in these families by comparing them with a healthy blood donor cohort known as interval, and some take-home messages from what Caitlin's been doing. So she definitely sees that rare damaging variants are over-transmitted from parents to children. The only families in which that doesn't happen is when the child has a diagnostic de novo mutation, which is kind of like an internal negative control within the study. That over-transmission is both from parents that clinicians have annotated as being affected and from parents who are annotated as being unaffected. And she's applied a framework that combines together de novo information from inherited information and case control information, and shows that does indeed start to, again, increase our power to detect these new disorders that we know are lie at the heart of increasing diagnostic rates. So that then brings to bear the idea that actually there are people who are, quote unquote, clinically unaffected in the general population carrying variants which, if we were to see in a child with a neurodevelopmental condition, we would be pretty sure were the cause of that condition. And there's some evidence from this from other work. So this is work from the Geisinger team and their Discover EHR cohort, where they identified just over 1% of their clinical population had either a CNV or an SNV that would be of diagnostic kind of confidence. But in that 1% of individuals, only a third actually had a neuropsychiatric diagnosis in their clinical record. 2/3 didn't. But they did also show that carrying one of these variants in that 1% did increase your risk of disease across a whole range of different neuropsychiatric disorders. And you can kind of see them down here. So this, again, is speaking to a somewhat broader clinical consequences of some of these disorders, and also still remaining questions about that are essentially unresolved with clinical data linkage about whether these individuals have been impacted by these variants or not. So another way of looking at this, rather than a kind of clinical population like in the Discovery EHR, is to use these big population biobanks. And here's some work done by Caroline Wright, taking a slightly more permissive definition of of kind of variants that we think would be pathogenic in a neurodevelopmental condition-associated gene, about 2% under these rules. And by working in a population cohort, one can start to see broader consequences of carrying these kinds of variants. So you're more likely to be unable to work, you're less likely to have a degree, you're more likely to score lowly on cognitive performance tests, you're more likely to live in a deprived area. That's what the Townsend Deprivation Index is. It has an impact on, negative impact on income, height, and BMI. And the conclusion, which I think is interesting to kind of read out, is our findings suggest that many genes routinely tested within pediatric genetics have deleterious variants with intermediate penetrance that may cause lifelong subclinical phenotypes in the general population. So to try and understand this a bit better, there's a limitation to what one can achieve by using adult cohorts. What one really wants to understand is longitudinally, what's the impact of these variants on child development? And so we've started work exome sequencing birth cohorts in the UK to try and look at the trajectory of these. And this is very, very early unpublished work, and just want to give you a kind of one particular kind of potential insight that's coming out of this. So that's the ability to contrast the impact of common genetic variation, which we know is the polygenic scores for educational attainment, cognitive performance, and so on, with rare genetic variants of the type that would be diagnostic in the kind of clinical context I'm talking about. Here, working with the children of the 90s cohort, one intriguing early observation is if one looks across IQ tests at 4, 8, and 17 years of age, the kind of negative impact of rare genetic variation appears to be decreasing over time. Whereas in contrast, if one looks at the impact of polygenic variation so here we're looking at the educational attainment polygenic risk score, its impact is increasing over time. So we don't fully understand this. We need more data to show it's fully robust. But it does suggest that this kind of longitudinal trajectories of different types of genetic perturbation may well be important to understand. And it may well be help to understand why some parents do not necessarily manifest overtly affected phenotypes when they carry rare variants that appear to be having a major impact in their child. And just lastly, in this kind of genetics phase, we've also looked again at polygenic variation. So this is definitely a clinical cohort recruited by clinical geneticists who suspected an underlying monogenic cause. We showed in an earlier study of a subset of the cohort that there was clearly some polygenic contribution. One could derive a polygenic risk score for presence in a neurodevelopmental disorder cohort. And that correlated in ways one would anticipate with neuropsychiatric risk scores and educational attainment risk scores. But Hilary Martin and members of her group have expanded this using data now from DDD and the Genomics England 100,000 Genomes project, and have made a number of kind of quite important, quite head-scratching, if you will, kind of observations. First is there's clearly more clear genetic correlations with polygenic risk scores of other neuropsychiatric disorders, including schizophrenia and ADHD. It's clear that the polygenic risk is higher in the undiagnosed children than in the diagnosed children, which one might expect, but we couldn't see before. One striking observation is that the unaffected parents have similar polygenic risk to their children, which suggests that at least some of this polygenic influence that we're seeing is not necessarily influenced in the development of the child, but in the broader environment in which they're brought up in. And there's a whole piece of work that has been developed in the context largely of family-based studies and population cohorts that is kind of speaking to this kind of what's often known as nurture via nature influence, that some of the alleles you see in the child are also in the parents and indeed in their parents. And one particularly kind of brain-scrambling concept is this is assortative mating. So this is when when people choose to have children with partners who are phenotypically similar to themselves. This is particularly strong for cognitive traits and educational traits. As many people met their partners at their educational institutions, perhaps not a surprise. But what that does is it introduces correlations between individuals and between different classes of variants that have the same effect on that trait that's being assortatively mated upon. So what that means in practice is that one individual who has a high polygenic risk of a neurodevelopmental disorder, that polygenic risk is correlated, of common variants, is correlated with the rare variant risk burden in their partner. So you can get tagging between individuals of polygenic risk and rare variants in their partner. So we need to bear that in mind when thinking about how we interpret some of these interplays of rare and common variants. We are in the foothills of understanding this, I would say. There's a lot more to be done. So I'm now just in the last part going to move on to the variants of uncertain significance problem, which I know many of you kind of recognize as being a big issue. So fundamentally, what we're doing when we do these gene discoveries is we're picking up that there's a particular developmental disorder-associated element, and at the point of discovery, we only know a little bit about that element. We know a handful of pathogenic variants and a handful of benign variants that we're confident are benign, for example, because they're in population cohorts and individuals, or they're very common, and in the population cohorts, they're in individuals who don't have the phenotype one would expect to see. But really, what we need to do is get to this stage where we know what every variant does. And one approach that I'm particularly excited by is this approach of using cellular systems and saturation mutagenesis to get at this. And this is really a cartoon. The way that data can often look is like this. One isn't necessarily looking at this from the DNA eye view is every single base change. The kind of protein eye view is let's think of every possible amino acid at every possible amino acid. Both are entirely valid and interesting. So these kinds of, there's a number of us have been really interested in these, and I know NHGRI has funded a group for in Seattle and Toronto doing this kind of work, and we've invested quite a lot in it as well. And I'm coming at this very much from the lens of clinical diagnostics in this context, but I just think these are incredibly powerful technologies, incredibly powerful data resources that will be transformative in a number of different areas. So, for example, our ability to use our genetic information in screening is greatly limited by our knowledge of what variants do. Our abilities to understand how proteins work will be enabled by this. I think it's really important to think about rare disease therapies. So I think having a variant effect map, where you know what every variant in a gene does, will become essential in pretty much any rare disease therapeutic development. It'll enable you to maximize the market size for that therapy. It'll give you confidence you've got the right people in your clinical trial for that therapy. And it'll give you insights into potentially mechanistic differences. Maybe there's more than one disease mechanism ongoing in your population. And so it would be important for tying genotype therapy combinations. And I think increasingly we'll see pharma companies being very interested, and we've already got a few interested that have been contacting us about having a variant effect map for their disease of interest that they're already investing huge amounts in therapeutically. But I'm going to come at this very much from the diagnostic lens. This is one really early example of this. This was from David Altshuler working on PPAR gamma, where they used a cDNA approach. So here they're putting in the protein in cDNA form, so they're able to look at missense variation, and they can clearly see that that missense variation, when it's having a functional impact, is very much focused on particular domains. So the DNA binding domain here and the ligand binding domain here. But one approach we're really interested in is actually, can we mutate the endogenous locus that will enable us to interrogate not just missense changes, but also enable us to look at deep intronic changes, UTR changes, understand when a synonymous variant might be influencing splicing. And so there's a very nice approach known as saturation gene editing where one can use Cas9 and homologous directed repair to introduce essentially any variant one might like into an exon or gene of interest, and read that out in a number of ways, the simplest being, does the cell grow over time? So, and famously, a great example exemplifying this technology was the work done in Jay Shendu's lab on BRCA1, focusing on the domains in which were already known to be diagnostically important in BRCA1, and showing really great performance, as shown here, in being able to distinguish between known pathogenic and known benign variants. But also being able to pick up things like missense variants that appear to be lowering RNA levels, so not having an effect at the level of the protein, but earlier than that. Synonymous variants also are having an impact at the level of transcription and splicing. So this was an approach we very much wanted to pick up on, and we wanted to use exactly the same assay, and we started with this gene, DDX3X, which is the most common cause of female intellectual disability out of all of the genes that we've been looking at in the Deciphering Developmental Disorders study. So it's a very simple approach. You go exon by exon. We include some intronic sequence. We make every single possible coding, SNV. We can make multinucleotide variants that basically introduce the same amino acid change, so we can have redundancy. We look at every indel that's ever been seen in any database. We can look intronically. We can look at UTRs. And this is work led by Lizzie, Honke, and Sebastian. So very simply, you're creating a library of cells. They've each got a different mutation. You see which ones die over time, because this gene, DDX3X, is essential in HAP1 cells. And so you're looking for the damaging variants that are depleting over time. So that's the cartoon version. This is the real version of what the data looks like for a couple of different exons. So here's exon 4. All of these depleted variants here are truncating variants or splicing variants. Whereas on exon 15, these mustard-colored dots are missense variants. So you can see there's a big cluster of missense variants that are depleting that essentially ends at this point here in the exon, which precisely corresponds with the end of the protein domain, the helicase domain. So this is what a map looks like of 12,000 variants across the whole gene. Again, at this level, one could probably not see very much, but hopefully one could appreciate the clustering of these mustard variants showing missense pathogenic variants are very clustered within the protein, as one might expect. So the first thing we did with these data was we did a time course. We looked over time at whereas the original paper only looked at before and after. And out of this, we could discern, using an unsupervised approach, 4 different classes of genetic variant. Those, the majority in green, that were unchanged over time. They just didn't change over the time course. That's this big green peak here. Those that were subtly enriched, that's this mustard peak. And then 2 classes of depleted variants, those that depleted very quickly and those that depleted much more slowly. So that was just a purely unsupervised approach. And if one just looked at synonymous variants versus nonsense variants, you can see there's a very clear distinction between the nonsense variants in red and the synonymous variants in blue, which is, of course, what one want to see. And one can see that as one looks at some of these more intermediate classes, like missense changes, about 15–20% are depleting. Codon deletions, it's about half. Essential splice, acceptor and donor variants, it's about 3/4 are depleting. And interestingly, both all the non-functional variants or the variants that change function are more highly conserved. They have higher CAD scores. They've got greater impact on protein stability. And interestingly, they're all depleted in UK biobanks, suggesting there's some selection there. Intriguingly, we have no idea what the phenotype for enriched variants might be. But it's clearly depleted, and they're clearly different from unchanged missense variants. So we saw a very low fraction of synonymous variants, about 1%, that looked like they were having an impact. Interestingly, given clinically, we often assume that variants that hit essential splice sites are pathogenic, and that's true about 3/4 of the time, but 1/4 of the time it isn't. We don't fully understand that. We found a 5' UTR variant that was clearly pathogenic, creates an upstream ATG. And overall, in this kind of this whole set of variants, about 3/4 of the variants that appear to be deplete are nonsense or missense, but 1/4 aren't. They're intronic, or they're splicing, or they're synonymous. So I think that's kind of quite sobering when one thinks about aiming to be complete in terms of diagnostics. If one looks at how these are distributed through the 17 exons of the gene, one can see the nonsense variants that aren't pathogenic are very much concentrated at the end, as one would expect to escape nonsense-mediated decay. The missense and codon deletions are very much clustered in these two big helicase domains. And there is some pattern to the splice variants that don't appear to be pathogenic in the last 2 exons in this exon 3 for reasons we don't fully understand. We can also get really nice insights into the intronic variants. So what I'm showing you here is essentially all of those introns for those 17 genes stacked on top of one another, aligned by the splice acceptor and donor sites. And you're seeing essentially all of here the depleted variants that sit outside of the first 2 essential splice sites and acceptor and donor sites here. And you can see, as expected, many of these are pyrimidine to purine changes in the polypyrimidine tract. So overall, about 6% of the variants we looked at appeared to be depleted in these introns. About 15% of the ones that were in this kind of donor +3 to +8 range, so over here, were depleted. And it also helps us to validate some of the AI tools. So Splice AI, which is a tool currently used, actually did a really good job of predicting these. Not as good as the data, but really quite good, better than I thought. So one of the things that also came out of this is, as expected, when we start looking at these unsupervised classes relative to clinical diagnostic classifications, that the pathogenic variants are very much that have been described, which may not be perfect in terms of their accuracy, but are very strongly enriched for the depleting variants. And the benign variant, so the UK Biobank and Nomad, there's a very large number of variants, essentially 0 of them are depleting. Intriguingly, this is a disorder that's much more frequent in females than males. It has been described in males, but actually, the evidence, male-specific evidence that these variants are pathogenic is pretty weak, and we think this needs more examination because we're seeing very little impact in a functional assay of those variants. Could be operating by a different mechanism, but currently we think that needs more work before it gets too extensively used clinically. So, of course, that's one way of doing it, that unsupervised approach. But we've got a huge amount of data to supervise and make most of the data to make a better clinical score. We've got 700 variants or so where we know what the right answer is, especially if we focus on the de novo mutations in females, which are much more likely to be pathogenic. So we can train a simple fat random forest approach, as everyone does these days. One can get a score that integrates data for across different time points. This is just showing one time point, showing really good separation of the functional and the functionally abnormal variants. And then one can start to compare that to the best in silico scores. This is quite a busy slide here, but I'll just focus on these red boxes are when it gets wrong. So 700 or so truth variants. There's one that the random forest gets wrong. And if one compares that to any of the in silico scores, they all get 10s wrong. The one that they gets wrong is a purportedly pathogenic variant defined by a clinician that doesn't look to be functionally abnormal in our assay. And if one were to apply the latest ACMG criteria, it probably wouldn't make it to likely pathogenic. So we think that one's an open question. So one can model what difference this makes in terms of criteria, in terms of diagnostic interpretation using the ACMG criteria. And this gives you a bit of a sense of how that works when one models this without the SGE data or with the SGE data. Essentially, the VOSs go down by 90%, and the proportion of variants that we know to be pathogenic or benign go up manyfold. So this is, I'm kind of giving you one example of one gene, which happens to be the one gene done in my group. There's another 3 genes we've done at Sanger using the same assay that Dave Adams' group has done, which have very similar kind of properties and accuracy. And we've generated partial data on about 20 or so other genes that are also essential in the cell line. There's there's several hundred genes that are known monogenic disease or cancer driver genes that are essential in the cell line. So we've got many more to go. How about using these data in real life? Well, within the DDD cohort, we don't have parents available for everyone. Where we don't have parents available, the 3,500 kids where we don't have both parents available, there's 23 kids that have a non-synonymous variant in DDX3X, and we don't know whether they're pathogenic or not. Applying these data very clearly bifurcates them into two groups, 14 variants that appear to be functionally normal and 9 that appear to be functionally abnormal. And if we look at those 9 variants out of 3,500, that then gives us essentially the same prevalence of disease as we see in the 10,000 trios. So that fits. So that's what I want to show you, just a deep dive on one particular assay, hopefully convincing you of the power of the data and why we're so interested in scaling this up and setting this up. But I think it's worth reflecting on where we are currently, because we currently have these clinical grade variant effect maps for a tiny fraction of the genes that we would wish to have them for. And I think it's worth thinking that this represents a logical third phase of human genome initiatives, from the sequencing to the association of functional elements with disease, to actually knowing what every single variant does in those associated loci. I just want to say big kudos to NHGRI for supporting a lot of this work, both for IGVF and the Atlas of Variant Effect Alliance. And I have to say, I've been involved in many different international consortia, and the Atlas of Variant Effect Alliance is just the most fun one that I've ever been involved in. I'm also like the oldest person in that whole alliance, which makes me feel a bit old. But it's an incredible group of really enthusiastic and really kind of there's a strong moral compass to that group and a real emphasis on open science, and I love working with them. But what we really need is a concerted effort on data generation that's commensurate with where we need to get to, and that's something that I'm sort of from my perspective at Sanger very keen to push on, work with partners, think about ways we can do it, and I'd love to, over the course of the next few years, think about how we really deliver this. I was intrigued as I reread before coming about the bold predictions, and number 7 caught my eye. Is that the clinical relevance of all encountered genomic variants will be readily predictable, rendering the diagnostic designation variant of uncertain significance obsolete. That's by 2030. So we better get our skates on, is what I took from this, because time is ticking. And so just some thoughts about how I can see this coming into being. So I think we're currently in this state where we've got these variant effect maps. Most genes don't have one. It's very sparse. I think a concerted effort that is a combination both of human genome projects like endeavors with multiple centers really scaling up production, but also democratizing the technologies so that many individual centers who might have specific cell biological expertise in particular assays can contribute as well. So I see this as kind of human genome project is left-hand side, few centers doing lots of work. PDB, Protein Data Bank, is kind of the right-hand side, lots of centers depositing. I see this as very much a mixed model here that will get us there quicker. I think that'll give us a much more populated atlas. And then I think will you be using AI and imputation on top of that in the same way that AlphaFold builds on top of Protein Data Bank to predict structures for all proteins. We'll be able to predict in a clinically useful way variant effect maps well before we've done every single gene in the genome. And if we do this together with our colleagues working in AI, we'll work out which data sets to generate in what order to get there quickest. So what do we need for success? We need diverse expertise. I think there's a lot of interest in industry on this, and we should welcome them in. I think we need to develop technology, but we also need to scale it. And the scaling, I think, is going to be a real challenge. It reminds me of the chutzpah of the Human Genome Project in imagining one could sequence the whole genome at a time when one's limited to 300 base pair segments of sequence. We need to democratize the technologies to make this model work well. It can't just be held in a few centers. And one of the things that makes me most optimistic is actually a lot of the data standards and coordination is far more in train than one would anticipate for the endeavor of this relative immaturity. I've been super impressed by the work that Doug Fowler, Alan Rubin, and colleagues have done with NHGRI funding to put together some of this infrastructure. And I also think there's a really good connections with the clinical community. It ties in rather than fights against existing guidelines and existing structures of people who are thinking about this. So I'm kind of optimistic about many of the operational logistical elements of this around the kind of hearts and minds pieces. Perhaps the biggest challenge is the funding and the scaling. So to conclude, international collaboration is going to be essential. We have to discover hundreds, if not thousands, of disease genes. Any one country can't do this alone. We have to do it together, and we have to find ways of making sure, as genomics goes into the clinic, that those data are not lost for research. We also need to understand the full phenotypic spectrum, and we'll only ever do that if we integrate data from population cohorts and clinical cohorts. Just looking at one in isolation doesn't give us that. And then finally, resolving the VUS problem is a tractable problem, but it needs concerted effort. And we have a good long history. We are both 30 years old this year, NHGRI and the Sanger Institute. And I think we should put our shoulders to the same wheel and make this happen. And so I've tried to highlight many of the people along the way who've done all the work, and I'm happy to kind of point out individuals down the line. But at this point, I'd be very happy to take questions or comments.
Questions for Matt, and if anybody in the room should come to the microphone, and if there's online questions, we can take online questions. So let Francis start us off.
Matt, that was fascinating in both parts. With the VUS analysis, it does seem that a lot of the genes where we would like to know the variant effect map may not be readily ascertained in a simple cell survival kind of assay. And that's one of the things that I've wondered. When you try to actually scale this to 20,000 genes, how many of them are going to have some very individualized assay in order to assess the effect of all possible mutations? Because that'll slow everything down, and you'll have to know quite a bit about the gene probably to design the right assay. What's your thinking about that?
Yeah, so I think it's a really good point. It's a key point. I think obviously cell essentiality in this mode is very much focused on loss of function mechanism. So there's both the underlying mechanism and the biological context. I think the good thing is that we have pretty good data for many of the genes that we care about in terms of truth data. So we can come at this from a kind of biological intuition, what's the right assay and the right cell type. But we can also come at this from a very agnostic position, which is, does it predict the thing that we care about accurately? And I tend to fall a bit in the latter camp. But I think what we do need is, and that's why we need that kind of mixture of large-scale production and smaller scale kind of group submitting, because I think there are absolutely going to be genes where there is one assay and it is in one cell type. But that expertise probably exists in the world. And we need to tap into it. We need to find how do we democratize the technology so those groups can be part of it and generate data of the same quality as useful and go into the same places as those large-scale production centers. I think there'll be a lot of intermediate. There'll be pathway readouts that can be used for multiple genes in a pathway, I would anticipate as well. But I do think we don't need to fetishize biological reality. So one thing that's interesting about DDX3X, for example, is HAP1 cell line is completely irrelevant pathophysiologically for a neurodevelopmental disorder. It's also completely irrelevant for medalloblastoma, which is what DDX3X is also a driver gene in. But that variant effect map works equally well for neurodevelopmental disorders and for medalloblastoma driver mutations. So
may that work more often for more genes. Like, second quick question. Already AlphaFold, of course, is able to tell you what happens to protein structure when you change an amino acid. So when you look at your data with DDX3X and just say, okay, how does the result compare with what happens when I ask AlphaFold to tell me, was that a significant change in the protein folding or not? How does the correlation look?
So Alpha Missense does a slightly better job than the ones that we had there, but it is nowhere close to at least in this gene, of the performance. So yeah, it's definitely an incremental positive step out from this sense, but it's and I think it's a hard job what it's looking to try and do, because one of the things that's the fundamental thing is that a lot of these algorithms leverage at a great degree conservation, and conservation is integrating selection across different environments, different mechanisms, different modes of inheritance, and so to have that level of specificity where you know that this set of variants causes the loss of function phenotype in this gene, whereas this set of variants causes the gain of function, and this set of variants cause the dominant negative ones, that's very hard to do from a conservation perspective because they're all just mushed together.
Sure. Okay.
Matt, there's a question online. I think I understand. I just want to make sure. So where does the effort to integrate patient data to solve the VUS problem intersect with problems of underrepresented or unsolved patient groups? So I think the question sort of getting at, in the process of looking at more underrepresented groups, does it give you insights that would be helpful to the solving the VUS problem?
Well, I would almost flip that around slightly. So I think one of the ways in which we will increase the equity of genetic testing is by having variant effect maps, because if we can interpret the data, which means we don't need to have the parents, and it doesn't matter if they're of African ancestry, then actually these assays will be leveling that playing field that's currently unequal.
But I guess for the question, they said to integrate patient data to solve the VUS. I'm not sure that's as much as what you're saying.
No, but I'm saying there's an intersection between the VUS part of the talk and the inequity of diagnostic testing. But I see this, a really powerful thing is actually these things level the playing field. I think it's going to be a very long time until we assemble together enough data to create variant effect maps out of humans carrying those variants. Clinically, clearly, it's going to take a very long time because only a subset of people have them. We know, for example, in DDD, 75% of the diagnoses we make are variants that have never been seen before in that gene. That's clearly going to be different in different disorders. And if one wants to use just human data alone to build up variant effect maps, one needs genetic data and really robust clinical phenotype data in tens of millions of people, probably hundreds of millions of people.
Carolyn?
So one of the things you talked about the variant effect, dealing with the VUS, but there's still the unknown gene part. And do you see, I can see a way that it's like, okay, well, if we do all of this, then we can kind of skip the gene part, perhaps. But what are you sort of seeing as the, sort of low-hanging fruit to help get that gene list more robust?
Yeah, so unfortunately, I don't think there's much in the way of low-hanging fruit. But I definitely see it as a two-step process, the gene discovery and the variant effect mapping. We've done a little bit of work that says, if you had a variant effect map of DDX3X, could you have discovered the gene quicker? And the answer is yes. It's not massive, but the answer is definitely yes. We've done that across a number of different genes now. So in theory, if we could actually generate all 20,000 variant effect maps very quickly, we would do a better job at discovering the genes more quickly. It's just at the moment, it seems like that's a harder ask than aggregating together the different clinical data sets. But if that does prove to be possible, it definitely will help. So yes, I do. We are very lucky because one of the issues with obviously clinical data is often more tightly constrained, is often more national in scope than research data. To do the analyses that I described of those 80,000 families, we're fortunate that the ethics and the consent and the fact we only need summary data from the two diagnostic labs mean we can put all of the data into the Genomics England environment because the Genomics England data can't move. All we needed was one of those centres to take the same perspective of Genomics England that our data can't move even in summary form and would be having a much more complicated. So I think it's that's the bit that worries me most is that we end up tying up our genomic data in clinical settings, not think about how it feeds back into the research domain.
Les, do you have a question?
Awesome seminar, thanks. For the Pakistani versus non-Pakistani, the graph was, I think, a little bit distorted because it was probably based on cases that you had, right? And so, because it looked like the de novo mutation rate of DDD disorders was much lower in that population, which it can't be, right?
Yes, so it's a relative rather than absolute, exactly. So what one can imagine is actually it's the same rate of de novo mutations, give or take parental age and potentially environmental exposure, but let's say it's the same rate. And then you've got the recessive added on top, which overall means a higher prevalence, which is what we already know about from parental relatedness studies as well.
2 to 3 times probably.
So it's a relative, not an absolute. So it's not like a decreased mutation rate.
Understood. And then your analysis of variant effect, hypothetical, where you implemented it as either PS3 or BS3, that's just the default. Did you actually calibrate it using the likelihood ratios from your data? Because your data may actually be quite a bit stronger than that.
Yeah, that's a good question. I'll need to go back. We have calibrated it. It is so Brunnock has become a verb in the UK, which is the first author of the paper about calibration of these things. So to Brunnock a map, we have Brunnocked it, but I can't remember if the particular analysis I showed is updated for where we got to.
Great, thanks.
Yes.
Thank you so much. My name is Java. I'm in Sean Budge's lab. So I'm just curious to know, because I see these analyses were conducted using GRCH38, right? And now we have CHM13, that is a complete human reference genome. And we see that with the T2T assemblies, that genome reorganization, which can also shift the SNP position in a genome. So looking at your slide where you talk about what are we missing, so do you think, okay, based on what we do in our lab, we see like the reference genome for zebrafish, there are some genes that we see today that are not present in GRCZ11. So my question is, do you think that the confidence that you have in the SNPs today or 5 years ago, is it different from the one that you can obtain from the CHM13 reference genome?
It's a good question. The short answer is we don't know because we haven't done the analysis. I would love people, so that all the data that I talked about, the sequencing data, they're accessible through managed data access. I would love people to take those data, the raw data, and actually do that question systematically. You know, map it against different reference genomes with different gene annotations and actually show what the added increment is. My gut feeling is it's going to be relatively modest, partly because one of the things we've done is we've taken, and anything you do will be somewhat biased, but there's 5,000 of these children which we've exome sequenced, and we've also run these array CGH, which have 5 probes in every exon, so we can pick up single exon copy number changes. And when we call CNVs from the exome data, we only miss a handful of them. So we've got an orthogonal technology that's picking up structural variants of the dosage type, and we don't miss very much when we call it from the exome data. That's not to say there might not be deep intronic variants or inversions that are balanced that we wouldn't see. But in the studies that I've seen where people have done whole genome sequencing in reasonably large numbers, those kinds of variants are pretty sparse in those, and are unlikely to be a major cause of the undiagnosed patients. But again, I'd really encourage you or your colleagues to kind of take these data and analyze them in different ways and show what can be done.
Okay, so number 2 question is, I see you prefer the use of GATK in calling variant. I don't know why you did not consider deep variant, because with the GATK you tend to get false variant compared to deep variant.
Well, I guess as you can imagine, over kind of 12 years of analysis, we've gone through multiple variant callers at multiple times and done multiple benchmarking exercises. And I think it's quite a number of years since we did our last benchmarking exercise, because at least for SNPs and indels, the margins were getting pretty small between them. So I can't remember if we included deep variant at some point, but we may well have done. But it's one thing we can see, for example, is that about 1,800 of the families we looked at were also sequenced and recruited to the 100,000 Genomes Project and whole genome sequenced. And so we started saying, well, what was diagnosed through one group and not the other group, given that's quite a big number. Generally speaking, the vast majority of the time, it's because a gene was on one panel but not the other panel. So it was all about, it wasn't anything about the variant detection, it was all about what gene was on your diagnostic panel at what time. And the other one, which is a smaller one, is things that were discovered in the genome that just weren't well covered on the exome. And so now it may well be, of course, they don't believe they used deep variant, but they didn't use GATK. They've used other approaches. But that was quite a fortuitous kind of by two different groups. Analysing quite a large number of the same families. And again, the kind of things that highlighted in terms of things missed by one and seen by the other were things that didn't speak to us about the real importance of using different algorithms so much as keeping your gene list up to date.
Thank you.
All right, well, I'm going to cut this off here because Matt's going to now spend a couple of hours, I think, talking to some of our extramural staff of NHGRI to really sync up what's going to happen. Probably you'll change hats a little bit for that. You'll take your hat off as an investigator and put your hat on as the Sanger director, and I know they're looking forward to that conversation. So I hope everybody will give one last round of applause for a terrific talk, and thanks for the discussion. Thank you.